Hello everyone, I'm Shiva and today I'm going to present a paper titled Analgesic Effects of Botulinum Neurotoxin Type A in a model of allyl isothiocyanate and capsaicin-induced pain in mice. Botulinum neurotoxin serotype A cleaves a SNAP, a specific protein belonging to the SNAP protein responsible for presynaptic neurotransmitter release. The cleavage of SNAP leads to the formation of a non-functional SNAP complex, which then blocks synaptic transmission. Botulinum has been approved as a prophylactic treatment for adult chronic migraine, but its exact mechanism is still unclear. Recent research indicates that botulinum inhibits the release of CGRP from trigeminal ganglion neurons and blocks the excitatory effects of capsaicin on brainstem sensory neurons. Capsaicin activates the TRPV1, a type of ligand-gated cation channel. TRPV1 channels are mainly found in small sensory neurons, where they play a significant role in pain and inflammation. Botulinum decreases the pain and inflammation caused by capsaicin by interfering with the trafficking of TRPV1 channels, which is achieved through the cleavage of SNAP protein. TRPV1, another a non-selective cation channel, is normally co-expressed with TRPV1. It's directly activated by AATC and indirectly by mediators of inflammation, eliciting nociceptor excitation and pain hypersensitivity. It's widely known that in addition to regulation of neuronal activity, TRP channels play a role in various non-neuronal processes, including endothelial functions. TRPV1 and TRPA1 are widely present in neuromuscular tissues, where they function as a vasodilator during neurogenic inflammation. The researcher considered the research question that what is the effect of pretreatment with botulinum on pain evoked by injection of capsaicin or AITC as TRPV1 and TRPA1 agonists near a neurovascular structure in mice? This study was conducted on adult male mice, C57, aged 3 to 5 months. Injection of botulinum in 15 picogram per mouse was performed subcutaneously in the inner side of the medial part of hind limb thigh, while saline was injected in control mice. Capsaicin or AATC were injected in the same site of pretreatment with botulinum and also into dorsal surface of the right hind paw at dose of 2 milligrams per mouse for capsaicin and 1% volume per volume for AATC. Capsaicin or AATC-induced pain was tested at day 7, 15, 21, or 30 after injection of botulinum in different groups of mice. And capsaicin or AATC were injected 1 hour before testing. The cumulative time that the animal spent licking, scratching, flicking, or biting the injected hind limb or hind paw was considered as a pain-related response or nonsensitive behavior. The locomotor function, coordination, and sedation of mice were tested by using an accelerating rotor. Rotor was conducted in 2 phases, habituation and trial. During habituation phase, mice were allowed to become accustomed to the stationary route at day 1, followed by further training for the next 2 days to a rotating route at 6 RPM. Training on the fixed route load was limited to 3 minutes over 4 consecutive days of testing with 2 trials each day separated by an hour. The mice were initially placed on a stationary rod for 30 seconds and then the rod began rotating at 4 RPM and gradually increased to 40 RPM over 5 minutes. Trials were stopped when mice fell off the road or after 5 minutes.
As a result, Figure A shows the duration of capsaicin-evoked nonsensitive behavior calculated in blocks of 5 minutes on the left and during total time of recording on the right in different group of saline or botulinum pre-treated mice. Injections were on the inner side of the medial part of the hind limb tight around the neurovascular femoral trunk. Capsaicin in saline mice evoked an offensive behavior that was maximal during the first 5 minutes and diminished during the successive time intervals. Pretreatment with botulinum reduced the total offensive behavior with maximal effect after 7 days. Analgesic effect of botulinum was still present at day 21. Post-hoc comparisons showed significant differences between saline and botulinum injected mice during the first 15 minutes for A7 and A15 mice, or the first 10 minutes for A21 mice. In panel B, capsaicin was injected into the dorsal surface of hind paw distant from the site of botulinum injection compared to saline botulinum injected mice. Both after 7 and 15 days of pretreatment showed not significant changes of capsaicin evoked pain response, indicating that botulinum didn't spread distant from the site of injection and don't inhibit the nociceptive fibers in the femoral nerve.
This figure shows the results of accelerated Rotaro test compared to saline. Both A7 and A15 mice don't show significant differences on the Rotaro performances, both during habituation and testing sessions. Here, figure A shows the duration of AITC evoked nonsensitive response in either saline or botulinum pituitary mice. Injection was on the inner side of the medial part of the hind limb type. Botulinum analgesia was maximal after 7 and 15 days, while it completely disappeared after 30 days. Post-hoc comparisons showed significant differences between saline and botulinum injected mice from 15 to 35 minutes for A7 mice, from 10 to 40 minutes for A15 mice, and from 5 to 15 minutes for A21 mice. In panel B, AITC was injected also into the dorsal surface of the right hind paw compared to the saline botulinum injected mice showed not significant changes of AITC evoked pain response at day 7 and day 15. Therefore, injection of botulinum doesn't act inhibiting the nociceptive fibers on the femoral nerve. As a conclusion, these studies data showed that pretreatment with botulinum reduced the nonsensitive response evoked by both capsaicin and AITC, and this effect was long-lasting. Therefore, these data demonstrate the inhibition of the expression of TRPV1 and TRPA1 by botulinum. In this model, botulinum doesn't appear to act on the near muscles nor on the nociceptive fibers of the femoral nerve. The absence of an effect on the motor innervation of the near muscles is proved by the normal performances during rotary tests. This article findings support the use of botulinum in the treatment of migraine, as previous research has shown that both TRPV1 and TRPV1 play a role in this condition.
Thank you for your attention.